My name's Jen Salerno. I'm an assistant professor in the Department of Environmental Science and Policy at George Mason University, and also part of the research group here at the Potomac Science Center, which is an off-site or another campus of Mason, is the Potomac Environmental Research and Education Center. So it's a group of interdisciplinary scientists or transdisciplinary scientists with background in biology, ecology, engineering, chemistry, etc. We all work together to address environmental research questions, particularly in aquatic environments. My laboratory is the Salerno Laboratory of Integrative Microbial Ecology, or SLIME, aptly named because many of the samples, or most of the samples and specimens we work with, are particularly slimy. We work on issues pertaining to coral health and disease, fish health and disease, biofilms in nature, and worked on microplastics in the environment. We also work in terrestrial environments, however, as well, including looking at the soil microbiome and how it relates to forest health. We've been working for a number of years now on a coral disease called stony coral tissue loss disease. And although we don't have tropical corals here in Virginia, we do our work based in Florida and Honduras. And so this disease showed up in about 2014 and has spread throughout the Caribbean, impacting over 34 reef-building coral species, and with a high mortality rate. And so a number of different scientists across the country, really, and the world, are working to try to identify the causative agent of this devastating disease. I think we're getting closer, but we still don't have one particular pathogen identified. And so that's been interesting in that it's allowed our research group to work with many different collaborators. It's also been extremely challenging due to the nature, the devastation that we've unfortunately observed in the field, but also, trying to mitigate the disease and identify the pathogen. But it's rewarding in that we've also become involved in a lot of coral restoration research and work, so that's a bit promising in hopefully restoring some of these impacted reef ecosystems. Another interesting project our laboratory is working on is a harmful algal bloom that's been ongoing in the Shenandoah River. So many of the PERIC members here are working on that collaboratively, which is a great opportunity for my lab and my graduate and undergraduate research students. So the bloom is a cyanobacteria, but actually likely a consortium of different cyanobacteria and other bacteria. So we're working to use molecular tools to identify the composition of those harmful algal bloom mats, to better understand their ecology, their distribution, what environmental factors influence them, what toxins they might be producing, and how this might negatively impact the surrounding ecosystem. We sort of study the unseen, right? The microbial majority in the environment, and although you can't see it, they play extremely important roles in biogeochemical cycling, organism and ecosystem health, so that's really rewarding. But I always tell my undergraduate students I had zero interest in microbes as an undergrad. It's something I've grown to really become interested in with time as I've progressed throughout my career. We have better tools now to study microbes in the environment, so it's a really exciting field to be in because there's so many technological advancements, as well as advancements in sort of the computation that we use to study our data, or to analyze our data. So that's really interesting as well. It allows us to collaborate with a lot of different partners, and especially within this department, since we do focus on not only environmental science, but also the policy aspects of things, we're able to take our work and try to translate it into action through policy recommendations, legislation, outreach, and so that's extremely rewarding because we can see the direct impacts of our work. It also allows sort of the training of the next generation of scientists, and our field moves pretty quickly in terms of technological advancement, so it gives students an opportunity to get hands-on experience working in the field, learning molecular biology, and also learning coding and things like that to analyze data. So a typical research day for me. So as you sort of progress in science, you end up spending probably less time in the field and here in the lab than as a graduate student, but I still do come in to work on projects and to train students, graduate students and undergrads. But a lot of that is guiding them in their research efforts. I typically have a lot of meetings, and I meet with my students regularly, both in a group setting like a lab meeting and one-on-one, to discuss their research projects, their progress, and their goals moving forward. A lot of emails, of course, but it's good because a lot of that is working to collaborate with colleagues on grant writing, on initiatives, on different projects, so it's rewarding. The nice part about it too is our research facility is located right on the Occoquan River, so we do get to be near some of our field sites, and it's a really beautiful place to work. And here we can interface with our colleagues and have discussions as well, face to face when we see each other at the Potomac Science Center. This is obviously a beautiful place to work. We have a nice new lab facility with all of the equipment that we need, access to shared equipment. I think one of my favorite places, honestly, is my colleagues, the camaraderie here, and everyone is very collaborative and willing to lend a helping hand, offer support, work together on writing grants and papers and doing research. We really try to complement one another's work, and so I find that's a really nice environment to work in. It's accessible. We live in a nice location, and it's relatively close to campus, but again, it allows us to have access to the water where a lot of our research takes place. So my lab has projects right here in the Potomac River, but we also do work abroad in Honduras, so we are able to do both local work and work abroad, which is really rewarding as well. So a cool fact is that we're all mostly microbes, and most of those microbes are doing good things for your body. So a lot of times people think of pathogens, but and that's true in nature too. Microbes are really important, a really important part of the ecosystem that can often be overlooked. And although you cannot see them without a microscope, they play very big roles, and they do look very beautiful under a microscope. My advice to students is always try to keep an open mind and think creatively. Like I mentioned, I wasn't initially interested in microbes, and the more I learned about them, the more fascinated I became. And one really cool thing about microbes is, I mentioned I'm not in the field and lab as much anymore, but the cool thing about studying microbes is they're everywhere pretty much. So you can go everywhere, and it's taken me to Antarctica, the bottom of the ocean, to coral reefs. So several times a year we do go down to the tropics and do scientific research diving. So that's a really exciting part of my job. And while I love being out in the field, I also love working in the climate-controlled laboratory, doing molecular biology, and also working with computer coding. So it provides a little bit of everything. So if you can, get a lot of experience to sort of find out what piques your interest, you'll find your path, and the more you broaden your skill set, that'll better prepare you for the future.
My name is Jordan Sims. I'm a fourth-year PhD student here in the Salerno lab. I did my undergrad at Rice University in Houston, Texas. I have a degree in ecology and evolution. Then came to Mason, joined the Environmental Science and Policy Department. So we're in a microbial ecology lab, which means that my research focuses on the processes that microbes are doing in the environment and how they're interacting with the organisms around them. So my research looks at how corals are interacting with microbes in, on, and around them on the reefs to cause different things to happen on the reef, like settlement, nutrient cycling, and those kinds of things. So I was really interested in studying microbiology in corals, which is exactly what Dr. Salerno studies, and is kind of what drew me to her. I reached out to her and was really fortunate that she was accepting students, and so she took me on to work on microbial ecology and reef ecosystems in Roatan, Honduras. So we do all of our field work there on the reefs. We have some great collaborators at the Roatan Institute for Marine Sciences, and we get to go diving and just get to see the reefs and be on the reefs, which is really exciting to do. I have a lot of different kinds of days as a grad student. We do a lot of field work, so we get to go out in the summertime to Roatan, and like I said before, lots of diving, being in the water, being on the reefs, working with our collaborators there. Those are some of my favorite days. When we come back to the lab, we have days where we're actually working in the lab, doing DNA extractions, PCRs, those kinds of things. And then we have a lot of data analysis days. So I spend a lot of days in front of my computer, coding, bioinformatics, analysis, that kind of stuff. And then writing also is computer days. One of my favorite things that I got to learn about after joining grad school was more about coral restoration, which is basically what you have to do after a coral reef is destroyed by whatever is destroying that coral reef, to rebuild it back up. We have our collaborators in Rotan are working on coral restoration practices, and we are trying to do science that makes those practices better. So I really love being able to learn more about management and restoration on reefs, but also how we can connect science with these real life applications. So it's really cool to be able to build our projects around the needs of restoration practitioners, so that we can make sure that our science is actually doing some good in the world. I think the biggest lesson I'll take with me after grad school is that science takes much longer than you ever expect it to. I feel like there are always delays at every step in the process, and it's just figuring out how to creatively work around them and push when you have the ability to push, to keep that science moving forward. But it's been such a valuable lesson, and Dr. Salerno is so supportive when these kinds of delays come up, that I learn how to take those in stride and be a better scientist because